Our next speaker in the last afternoon session is Qifa Zhang from the National Key Laboratory of Crop Genetic Improvement at Hua-Song Agricultural University. Qifa has really been, he doesn't need much introduction. He's been working in rice genomics and genetic improvement for many years, but just to see how busy he is and what has come with his successes. He's now director of the National Key Laboratory of Crop Genetic Improvement, the National Center of Crop Molecular Breeding, and the National Center of Plant Gene Research. And you're on the editorial board of many journals, including the Annual Review. And he has also a lot of accolades. Too many to mention here, but just a few. He's been elected to the Chinese Academy of Sciences, of course, but he also got the Alumni Award from UC Davis, and he's become the Foreign Associate of the US National Academy of Sciences. So, Qifa, we are looking forward to your talk on reproductive isolation. You are not isolated.
Thanks, Willie, for the organizing such a great event, and also it's a great pleasure for me to be here. So that's my second time visiting this campus, this city. And what I'm going to talk about today is something new outcome from a lab, understanding reproductive isolation based on a rice model. So no need for introduction. Reproductive isolation is awfully important in plant biology, but probably you don't know the importance of reproductive isolation in plant breeding. So there are two rice groups, two groups of rice, two groups of whatever you call in cultivated rice, a species or Rhiza sativa. So one is called, the two groups were recognized very long ago, at least 2,000 years ago in China in the Han Dynasty. So one was recognized as Xian, and the other actually should be pronounced Gen, but at that time it was a Ken placed there. And then these two groups later on were labeled, one as indica, the other as japonica. So we could see a lot of genomic diversity. Whatever trait you are looking for, you could see differences between the two groups. So very recently, Bing Han published a lot of sequences, so we could see very clear separation of the two rice groups, indica and japonica. And also, of course, some intermediate. So if you make a hybrid, just taking a random sample from indica and japonica, this is what you are going to get. So you could get a lot of heterosis. Like this one here, Libombara is something very familiar to the rice community. This is a sequence genome. The other is indica. So that is a parent of a very popular indica hybrid in China. So make a cross between these two, we get a panicle probably twice as much as the parent. So we see very high heterosis, but very low fertility. If you look at this very carefully, you see green ones and yellow ones. The yellow ones are filled greens. So the green ones are empty, spicular. Very low fertility, and also that's a great interest for utilizing interspecific heterosis. And then we have problems. So to evaluate, to see the extent of such a problem, we made some sort of analysis, taking a diversity of germplasm with representatives from many countries. So we made a diallele cross to see the extent of such a hybrid sterility in rice. And then this is what we see. So we see we have indica by indica, of course, japonica by japonica, indica by japonica, or japonica by indica. So we see as low as 0% of fertility, seed setting. So that's basically completely sterile. And then, of course, indica by indica, japonica by japonica, those are perfect, right? Those are varietal crosses within subspecies. So those are normal. So we see anything between 0% to something typically like 50% of fertility. That's for the intersubspecific crosses. And then a lot of problems. If we look at the hybrid cytologically, we could see male problem, we could see female gametes abortions, we could also see reduced fertility between the uniting gametes at the time of fertilization. And then if we do sort of a quantitative analysis to see the extent of the problem and the contributions of the two components, contributions of the female component, the correlation is about that in this case, the correlation between embryo sac fertility and spiglet is 0.6. Correlation between pollen fertility and spiglet fertility is 0.6. So it's about equal contributions if you make a cross, not just a random cross between typical indica and japonica varieties. Then there were models proposed early days in the analysis without data support, without really seeing anything genetically. That were two very early models proposed in the 1950s and 1960s. So one is called duplicated lethal model. That's alleles interacted unfavorably to reduce fertility. The other is single locus allelic interaction model. So that could be the interactions of alleles at single locus to generate negative interaction to reduce fertility. And over the years, there are a lot of studies, genetic analysis and mapping, seeking for germplasm for fertility, to understand the extent of fertility problems. So overall, in the literature, we could see something as many as 50 loci were mapped for any degree of fertility problems in the sort of indica japonica hybrid. There's a third group called wide compatibility. Wide compatibility was first discovered in 1939, very, very many years ago. So from Java Island. So a group of rice varieties that could be crossed with both indica and japonica could produce normal hybrid, no problem. And then the wide compatibility brought hope for the breeders, right? Could overcome the fertility problems, making use for breeding, for bridging the sort of fertility barrier between the two subspecies. So this is something we see in the, if we make crosses between pairwise crosses of the three varietal groups. So we have this one here as indica, this particular locus I'm going to talk about in detail. So one group called indica japonica wide compatibility. If we make a cross between indica and wide compatibility, they are fines, and the hybrid are fertile. And then make a cross between indica, between japonica and wide compatibility, and their progenies are also fertile. And only in the case, if you make a cross between indica japonica, we have a hybrid showing very low fertility. And so over the years, people are seeking, try to understand how the wide compatibility is controlled genetically. So it's a sort of cross called three-way cross design, which goes like this. Indica by wide compatibility variety. Wide compatibility variety could be phenotypically looking like either one or the other. And then get F1. F1 is crossed to another type, indica wide compatibility japonica, or the other way around. And then get in the progeny and do the progeny test to score, to examine the fertility of the progeny by whatever, spiglet fertility or pollen fertility or embryo sac fertility, depending on your ability to evaluate. And then later on, using molecular marker linkage map to evaluate this. So in one of the crosses we have been working on, this is something we got. We mapped one locus. Actually, I got ahead of myself. This locus appeared here in this crosses. In this cross, we mapped this locus called S5 on chromosome 6, although also some other minor effect on other chromosomes. And then we sort of used different germplasm to do very much of the same thing. We mapped loci controlling indica, japonica, and white compatibility in the germplasm, the loci. So again, we identified the locus at the chromosome 5 playing a very important role in controlling hybrid fertility. And then another cross, we sort of dissected the effect of pollen fertility, embryo sac fertility, and spiglet fertility. We see the contributions of pollen fertility, embryo sac fertility, sort of co-localizing at also the S5 locus. Then we focused our effort at the S5 locus, and then developed many near-isogenic lines. So transferring the locus to different genetic background. So from one parent to the other, and the other way around into indica background, into japonica background, white compatibility background to see what would happen. And eventually we mapped this using near-isogenic lines, using embryo sac fertility, mapped the S5 locus to a fragment of this size. I cannot, 40 kb, okay, 40 kb fragment containing 5 open reading frames. At the same time, many other Chinese groups are also interested in working on this, and also published about the same time another mapping result. So these two results overlap by something like 20 or 24 kb, if I remember it correctly. So with 2 open reading frames overlapping in this region. So at that point, we studied our transformation. So I said 2 open reading frames. We started with 3 open reading frames, 3, 4, and 5 in our numbering system. So the strategy is like this. If this is a correct candidate, if we transform the one from indica into japonica background, that just happened that way because japonica is much easier to transform. And then 3, 4, and 5 from indica into japonica background, if we got the right candidate, it should show reduced spiglet fertility and also embryo sac fertility with normal pollen fertility. And then we did 3, 4, and 5. We saw significant differences only with the transformant of OF5 in T1 generation. And then, so not OF4, OF3, OF4. And then this is something we saw of the transformant. So the pollen is completely normal. We saw embryo sac abortions of the transgene positive plants, and also the fertility reduction, almost nothing. No seed setting in the transgenic plants. And then that's a numerical statistical test of what we have seen using T1 generation to do co-segregation analysis. And then that is something we saw the effect of open reading OF5. So at that point, we sort of did the bioinformatic analysis, and then OF5 corresponds or encodes, or is predicted to encode something called aspartic protease. So it has high similarity with aspartic protease. So this is the structure of the genes. It has a signal peptide and also 2 active sites of the aspartic protease. And then if we compare the 3 groups, indica, represented by Balila, and represented by Nanjing 11, japonica by Balila, and white compatibility, the 3 alleles, we saw the differences. So indica and japonica differed only by 2 nucleotides, one at here, one at here. So both created amino acid substitution. And the so-called neutral allele, wide compatibility allele from this variety, or allele from wide compatibility variety, has 150 amino acid deletion, 115 amino acid deletion in a signal peptide. So cause mislocalization of that particular protein, and that is something we interpreted as the loss of function mutations at this locus. And then we did transformation of the indica allele into the wide compatibility variety. And it behaves like an indica, not wide compatibility anymore. So that is one episode of the story. So the feature of the open reading frame 5, encoding aspartic protease, predicted proteins of indica and japonica alleles differed by 2 amino acids, and the neutral allele has a deletion of signal peptide leading to mislocalization of the protein. So the question now is, how can we explain the hybrid sterility by a single open reading frame, which is OR5 in this case? So that is very difficult to explain. Then we proceeded further to look at the progeny generation of the hybrid. Then indica vanilla, japonica vanilla with the indica, that's at the background of neoestrogenic line. We saw in the progeny BC2, we saw segregation distortion. So it significantly deviate from the expected 1 to 2 to 1. And then that is the background of neoestrogenic lines. If we make a cross just between the variety, near 1911 and vanilla, we also see significant deviation from the 1 to 2 to 1 expectation, and other crosses also showing the same. So this locus and typical, this locus showed segregation distortion in both isogenic background and also in typical indica japonica hybrid. So segregation distortion is sometimes or always or most frequently or in the literature generated by more than one locus or several genes working together, or at least 2. So if we, as we said, if we go back to look at our mapping result again, so we could not from our own data rule out the possibility in this region more than one open reading frame or genes involved in this system. So in our transformation study, we took OR5, OR3, OR4, OR5, all from indica variety, and then did transformation to japonica. That is our expectation. And then, but whether that is something we did correctly, then we went back to check to compare the sequences of all the 5 open reading frames. We detected predictable functional differences in OF3, OF4, of course I said OF5, but not OF1 and OF2. And then we are sort of based on the sequence analysis, we assigned the sort of symbol or just sort of designated the alleles from different varieties as like the one, OF3 from the japonica one as OF3 minus. I have reason for that. The wide compatibility variety as OF3 plus, Dula, OF minus, and OF4, the other way around. So the one from Dula give you the plus sign, 1911 a minus sign. OF5 already showed we have 3 alleles. And that's, we did a lot of genetic analysis, which is a little bit difficult to explain. We saw a lot of the very severe effect. OF5 by transformation would kill the gamete. So I showed that, which particularly killed the embryo cell. And also, if we transform, we sort of did this and did the comparisons of this one. If we have OF5 for sure would kill the gametes. And then at the presence of OF3 if we have OF3 present, if we compare this one and this one. So in this background, the fertility is 46%. So if we add the OF3 to it, it's rescued to 75%. That means OF3 has a rescuing effect. If we compare this one, this tells the same story. So we have OF5, Balila has OF4 plus, and then we have OF3 plus transform, also could rescue the gamete from at least partially, so significantly different. So that means OF3, the conclusion from this table is ORF3 has a rescuing effect on IJ hybrid fertility. That's one thing. Another is we compare the effect, the same shade, the genotype with the same shade. We see very much different. So those are the same. The three haplotype composition of this one and this one are the same. But with the presence of OF4, it kills a gamete from 79% to 39%. So if we compare this one and this one, again, the same story, and this one, the same story. And which means that the OF4 is also important for killing. So OF4's presence of OF4 together with OF5 also kills the gamete. So without too much of the sort of genetic data comparison, basically this is a conclusion we reached at. OF5 initiated the killing, and OF4 is a partner of the killing, and OF3 protecting the gametes from killing. So this is the drawing from a student. So OF5 is the arrow, and OF4 is a bow. So OF5 together, this sort of OF4, and kill the gamete. But OF3 provide the shield. So with a good shield, the gamete is protected from killing. And with a broken shield, the gamete is killed. So that is a system. So we are sort of evaluate the expressing of the genes and express at very low levels throughout the life cycle, except at the stage of female gamete genesis. And then we sort of localize the proteins, determine subcellular localization, basically of. I said OF5 already. We showed it was localized extracellular in the cell wall. And OF3 localized in ER. So OF4 localized in plasma membrane and also Golgi body. And then we come up with a model, genetic model. We make a cross between 1911 indica and Balila japonica. And this is a hybrid. The hybrid would produce OF5 plus and minus, and then OF4 localized in the membrane, and then OF3 localized inside the cell. So the good OF4, which is wild type, would be functional at the membrane, and then the mutant would not be localized at the membrane. And then that is the at the time before meiosis or the hybrid, or at the time of meiosis. And after that, in megaspore formation stage, and then these two would separate. That is after meiosis. So this one has a good OF3, although OF5 would initiate the killing. OF4 localized at the membrane, OF3 could undo their effort, so protecting the gamete. And the later stage, the same thing, the gamete would be fine, viable. And then another gamete segregated or derived from this hybrid, the one that's a typical japonica with a 4+, with a membrane localized functional OF4. And then at the phase of OF5, initiate the killing process. And the earlier stage, OF3 would be okay and protecting the gamete. And later on, you run out of OF3, and then the gamete would be killed. So the gamete is abortive. That's basically a sort of model we derived from this sort of analysis. And then we sequence a lot of germplasm from the gene bank and detect a lot of polymorphism. in the rice germplasm collection. Basically, this sort of genotype will recover from the sequencing. So 3 plus plus plus, all those are plus, and this is the most prevalent one. That's very interesting. It could kill, it could protect, and it still is the most frequent one. So that is probably, we have an outgroup. So that suggests this one is probably the ancestral type, the wild type. Gradually, it lost one or the other, and then become some kind of mutant. But the suicidal combination didn't appear. That is something for sure zero didn't observe. So the other ones, just one sentence, conclusion. So this sort of observation agrees with the model we proposed. And then we did microarray analysis to try to figure out what is really going on. We found the overexpressing, the induced expressing of OF3 in the presence of transgenic OF5. And then we did a lot of analysis, and then we identified ER stress-induced genes all showed induced expression in the presence of OF5 in the background of Barilla. And then also induced at a later stage a bunch of PCD-related genes. So this is, and then we also checked the PCD processes in this, using tunnel analysis. And then the conclusion is that with OF5, transgenic OF5, which resulted in gamete abortion, and the PCD processes occurred earlier and also lasted longer, at the very end killed the gametes. So this is a sort of a hypothesis biochemically, and it has some sort of, basically this is summarize what we have seen here. Of course, still at the stage of hypothesis. So some sort of substrate, and then become some sort of product because of OF5. And then the product, when the OF3 is wild type, it is okay. The cell would not have any problem. And when the OF3 is a mutant, and it cannot undo the effect of ER stress, and at the very end, it causes PCD and embryosacral abortion. So summary of the killer protector system. It has involved 3 open reading frames that are very tightly linked, only within 100 kb genome region. And during female gametes, during female sporogenesis, action of OF3, OF5, and OF4 together will cause embryos ER stress. And OF3 could prevent ER stress and produce normal gametes, or otherwise the gamete is killed. So resulting in preferential transmission of OF3 gametes result in segregation distortion. And a couple of other stories generated from rice very recently. One is rice pollen hybrid incompatibility caused by reciprocal gene loss. So there are 2 genes encoding the same protein. So located on different chromosomes, hybrid is made in this way. The pollen carrying mutant from both loci would be sterile. So that is one case, duplicated lethal. Another also cause pollen sterility resulting from mitochondrial genes, nuclear localized mitochondrial gene, mitochondrial proteins. So again, duplicated lethal when this the mutant form meet in the gamete, that result in sterility. That's one. Another case. Another case is hybrid male sterility in rice controlled by interacting between divergent alleles of 2 adjacent genes located, again, next to each other. So this is a model. I'm not going to details. So that comes back to a very, very early proposals. Everybody is citing the model, Dubzhansky model for reproductive isolation. So basically, this is what they said. starting with AABB, but at that time they didn't start with capital letter, they started with a lowercase letter. And gradually you evolved into capital letter in one of the clades, another one, capital B in other clades or in other lineage. And when these two meet later on, and the interaction between the capital A and capital B produce isolation mechanism. So that was the model produced 80 years ago. So based on the rice analysis, I think we could propose three models. So parallel divergence model, sequential divergence model, parallel sequential divergence model. So the parallel divergence model, starting with a single gene appear in a diploid genome and then get duplicated and then diverge in two ways. In one lineage, this one changed function. The other one, in other lineage, this one changed function. When these come together, they cause problems. That is one way diverge in parallel in the process of evolution. Another is sequential divergence. Sequential divergence, and this one remained the same. But this one, like in our case, in the case of one of the rice example, and the A diverged, and then at the very end, B diverged sequentially, and then when they meet, they could cause problem because the original A would negatively interact with the mutated B. So that would cause problem in the very end with the gametes. So another model is a parallel sequential divergence model. If this one goes this way, and the parallel divergence first, there's three genes. One could go this way, a B mutated, and this lineage A mutated, and then further, when this mutated, allows the other to mutate. When they come back, they produce some sort of killer protector, as I described. So basically, the data accumulated so far can be classified into one model or the other in rice, and also a number of animal studies. So from reproductive isolation, rice, Arabidopsis, Drosophila, the examples conform to parallel divergence model. Examples conform to our sequential divergence model. So the one agrees with our proposal parallel sequential divergence model only this particular case, S5, in the case I just described. So the perspective, I believe in rice, a lot of genes will be cloned in the future years, in the next few years, for reproductive isolation, especially for indica, japonica, hybrid sterility, and some other type, hybrid weakness. And cloning and molecular characterization of this sort of genes would uncover diverse mechanism of reproductive isolation for evolutionary understanding of the systems. And this will understanding and this effort will greatly facilitate development of intersubspecific hybrid. So that could be very, very useful. And also it could promote reciprocal utilization of rice germplasm in rice breeding of both subspecies. So many people, actually this work was started, I first made the first cross in 1990, so 22 years, so many, many years. Many students and also our collaborators involved in this project, also supported by a number of funding agencies and programs. Thank you very much. Any questions? Get to.
Shifa, thank you very much for the tour de force through rice genome evolution. That was great. So, questions? Maybe I can start with the first question while still everybody recovers.
It's quite complicated. It's my first time to talk about this.
So the aspartic protease, right? That's one of the genes. Do you know actually the function of the two other gene products?
Well, one of them is a BIP, OF3.
Yeah.
ORF3 is a BIP, but it's not complete. even the, I put a plus, if we align it up with a functional BIP, it's missing something.
And that BIP is located in the ER.
Yeah, right,
yeah.
right.
But you think it's non-functional?
Well,
but is it a substrate?
One is functional. Even the functional one is not complete.
Yeah.
The other one missing even more.
But it's not a substrate of the aspartic protease.
No.
So what's the third gene?
The third is a membrane localizing protein without non-function.
So
okay.
There's still some work to do.
Yeah, more to do, right.
Okay. Other questions? So what was the situation? I mean, I didn't get it that quickly, but what was the situation in Dula? Because that's a wide compatible.
Yeah, Dula is regarded as a wide compatibility variety.
Yeah.
I believe it's 3. It has minus, minus, plus.
Minus, minus, plus.
Right. The 3 OF3 is a plus.
Okay.
You are familiar with Raytheon plasma. Huh? Dula, yeah, it's so-called Aus-group variety.
Okay, yeah.
So it has a 3 plus, 4 minus, 5N in our classification.
So it would be interesting to look at actually functionally why, when 2 of the components are missing,
it's still okay. It's a rice variety.
Yeah.
So it's a rice variety widely used in breeding programs, although it does not appear to be a very healthy one.
But it has a lot of interesting characteristics, right?
Right.
Okay. Question.
So, Chifa, when exactly does abortion occur? Does it happen immediately following meiosis in the megaspores, or does abortion happen only later during embryo sac development?
I didn't get it.
The time point of
abortion.
Embryo sac abortion.
The time point of embryo sac.
Time point of embryo sac abortion is, well, obviously after meiosis.
Immediately following meiosis.
After meiosis, we know the stage. I don't remember it.
Okay.
I have a question about the wide compatibility varieties. The Aus group varieties like Casalat and Dular, they're considered to be more close to indica types. So do you see a difference that when you cross these varieties with indica types that the fertility rate is higher as compared to when you cross these two Japonica varieties?
I didn't quite get it.
So the fertility in Casalat, what was the other one?
I can repeat that.
Casalat.
Casalat and Dular, both of them they belong to Aus type rice varieties.
No,
so we were just discussing.
I'm not sure. If we make a cross between Casalas and Nippon Ba,
yeah,
that would be probably, I would consider as an Indica Japonica hybrid.
Yeah, that's what my question was. You had a slide where you
we have to check the genotype, like in terms of our finding. We have to check the genotype, what's the genotype, haplotype, 3, 4, 5. So that prediction may not be 100%, but it should be very accurate.
Any other questions? Yeah, there's one more question and then we'll move on.
I have a bit maybe a stupid question. So if you now add ORF3 into Indica or Japonicum, can you now make hybrids and use it for breeding in the end?
Again, I didn't get it.
So if you put OR3, let me see if I got it correctly. If you put OR3 into Indica or Japonica, can you then use it in hybrid breeding?
Yeah. Well, basically, if the breeder is transferring or introgressing, like what we have been doing in developing neoestrogenic lines, so it's a transfer is a big chunk of the fragment. So not only one of them. And its function, well, sometimes, very frequently, it didn't come in a way as expected when doing a screening by the breeders, because they didn't know there are 3 open reading frames. And in some of the crosses, they see, in some of the Indica Japonica crosses, they could work. In some other crosses, they don't work. I didn't know that. So nowadays, we need to sort of genotype the parental lines or whichever the donor lines to see the genotype, so that we could use them efficiently as wide compatibility.
Good. Thank you very much again, Chifa.